marks are easy to get. So we shall see how we're gonna get these 27 marks. Meiosis, there is some people they call it a devil, but it's not a devil if you really attend our classes. So it is 21 marks. So having these two topics is enough for you to be skipping out of out of what? I think you'll be able to skip out of um the low mark which you call the 30%. But I always tell my students, please don't be fooled by any system. I'm saying it right, I think. By any system, 30% is not taking you anywhere. So make sure that you focus for something above 50%. That's why I'm saying that please, 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 please read more, focus on more on this. You'll be able to get more than 50%. Genetics, yeah, we look at where to get free marks in genetics, and then that is 32%, which is 48 marks. So if you have genetics, as genetics is enough for you to get out of the level one based on the government level one, which is 30%. 30% we only need 45. So however, to me, it's not a pass. You are still failing, but we want to achieve so that you push these marks more than what? More than 50%. Then evolution is divided into two. That is evolution by natural selection and human evolution. We're going to see where to focus on when it comes to human evolution and when it comes to evolution by natural selection. Guys, are you ready? Let me know if you have any challenge. Junior is saying, hi, it is not. It Hi, is it on my side? How about it? I don't know what you're saying, but let me know if you have any challenge so that I'm able to sort you out. Yeah. By the way, it is a live class. However much today I'm not putting my face on. Yeah. But if you want, if there is any need, if you want to emphasize something, then automatically I'll put that face on. Guys, DNA code of life. I'm not going to be teaching. I'm going to highlight where you're supposed and then remind you. That's it. And then I show you that this Zwan is saying challenge on human evolution. Okay, I'm going to reach there. Challenge on human evolution. Don't stress. I'm going to reach there. They're saying that how did I get distinction? How do I get distinction material you order from us? All right, let's start. DNA. Here on this diagram, what you need to know is what's called the chromatin network. Yes, chromatin network, then the nuclear membrane. Please make sure that you know that. Then here, when it comes here, describe the structure of DNA, 6 marks. Yes, describe the structure of DNA. This is the reason why we have all this. Ne? Yes. So here, these are nucleic acid, whereby you have messenger RNA, transfer RNA, ribosomal RNA, in actual sense RNA and DNA. So all of these are made up of nucleotides. So the building blocks are called nucleotide. The building blocks of DNA, if they ask you that, you say it is a nucleotide. Sometimes we call it a monomer. Please don't forget about that. Ne? Done. Building blocks are made up of a phosphate, sugar, nitrogenous base. If they ask you about DNA, then make sure that you know that when it comes to sugar, don't say sugar, say the deoxyribose sugar. And then when it is RNA, be specific. Don't say that sugar. No, say that ribose sugar. I think I'm clear there. You need to know the symbol that a circle means that. This pentagon means that, and then that square means that. Is it clear? Yes. Then we go to types of DNA. Here the question comes like this. Let me tell you how the question comes here. The question comes here, give 2 places where you can find DNA in the plant cell. So you'll find out that people won't talk about the mitochondrial DNA or they won't talk about the nuclear DNA. In the plant cell, if they bring about plant cell, you mention all of them. nuclear DNA, mitochondrial DNA, chloroplastic DNA. But if they say the place, then you talk about nucleus, mitochondria, and chloroplast. I'm done. Let's go. When it comes here, what you just need to know two things. Who discovered the structure of DNA using X-ray diffraction pictures? Only this now. Then that's when you talk about Maurice Wilkin and Rosalind Franklin. If they ask you who discovered the structure of DNA, just like that, you talk about James Watson and Francis Crick. The last question is who discovered the structure of DNA using the X-ray diffraction pictures? Sorry, who discovered that DNA is double helix with the complementary base pairs? That's when you talk about James Watson and Francis Crick again. So I think, guys, you understand that very well. Let's go to the next sub question. This one can come as 6 marks. Describe the structure of DNA. I don't need to memorize what is here. Yes. However much you need to know the marking points, what you are looking for. Is it clear? What you are looking for. So, what you're looking for here is one thing. Come back to the structure of DNA. Yes. Go to the structure of DNA or draw it. Okay, I'll come back here. Draw the structure of DNA and then start to describe it. DNA is double helix with the complementary base pairs. It's made up of nucleotide. Each nucleotide is made up of each nucleotide is made up of phosphate, sugar, nitrogenous base. Then you have the nitrogenous bases are adenine, thymine, guanine, cytosine. Adenine is complementary to thymine, and guanine is so you explain what is the structure of DNA. If they ask you describe yourself, you tell us you are big, you are small, you are dark skin, light skin. What you see, that's what they want. Please, you talk about anything you know about DNA. And these questions, make sure that you just give everything you know. Don't just list and then say, maybe this is wrong, this is right. No, you just. Someone is saying, I speak too fast. No, the reason why I'm speaking too fast is I'm assuming that you guys you know this DNA. I want to put more emphasis on these other topics like evolution and the genetics. However, if you see that I'm too fast, let me know. If okay, let me just reduce the speed. I'm sorry. I'm sorry. I'm sorry. You call yourself who? Cupcake. Cupcaker, all right, and the B-boy, right? Hmm. You're saying so describe that structure. I think I'm done there. What is the function of DNA? Basically, it carries genes for inheritance. Yes, and also makes it to provide information for formation of a protein. Is it clear? Yes. All right. Know the functions of DNA. Now, when it comes to base calculations, guys, I think you know the base calculation. We have like 2 ways or 3 ways in which we can bring this question. If adenine is 20, I'm just giving what is the percentage of thymine? Or what is the ratio of thymine to cytosine? Let me just do like that. I'm looking for the ratio of this to this. So if adenine is 20, adenine goes with thymine, cytosine goes with guanine. Whereby if adenine is 20, obviously this is 20. So it makes me to have 40. So 40, remember it's percentage. So minus 40, so I'm gonna get 60, and then 60 is for these two, meaning that if I have 60 divided by 2, I'm gonna have 30 here, I'm gonna have 30 here. So since I've got these two, then automatically I will come here and say this is thymine is 20 and then cytosine is 30. Then I divide by 10 so that I make them to be simple so that it gets into 2 by 3. So that becomes the ratio of thymine to 1 to cytosine. I'm just doing this so that you are able to remember what you did before. All right, we go to the next question. When it comes to DNA replication, please don't go without knowing these questions. What is DNA replication? Process where DNA makes the exact copy of itself. When does it occur? It occurs during interphase or before the cell divides. Where does it occur? It occurs in the nucleus. Why does it occur? This is the importance. Why is it happening? This is the what? The importance. Palisa, you're saying I should start again. Palisa, I can't start again. I've just started it. Just continue. You replay the video at the end. All right. Thank you so much. They're saying that double the chromosome number. It doubles the chromosome. It helps to double the what? The chromosome number. So meaning that the results in this information. So basically, why do we double the chromosome number? So that when it undergoes meiosis, we are able to to have each cell is supposed to have same number of what? Chromosome. Sir, do the calculation. Again. Renato. Yeah. You are seeing the calculation for the first. Oh my God. Hi. Honestly, just to give you a different example. So you have to know these questions. You understand? You have to know these questions. You can't go without knowing. Some of the questions they bring in the DNA describe the structure of DNA, DNA replication, transcription, translation. Those are the long questions they can bring in the DNA. All right. Guys, please take note of the information you want me to talk about maybe at the end. Ne? Yes. All right. Then, so when they say, how does it occur? Let me tell you something, guys. When they say, how does it occur? Yes, I'll do the genetics. Don't stress about it. How does it occur? You describe the process of DNA replication. But sometimes we can bring something. Let me see if I did prepare something like this. For example, like this. This question is saying that the diagram below represents the process that occurs during protein synthesis. Identify X, identify Z. But at here they say that name and describe the process. Sometimes they don't say name the process. They just say describe the process. So meaning that when you name the process, we give you marks. And then when you describe it, we give you marks. Even if they have not said that name, as long as they have not identified that process, make sure that you identify it and then you describe it. Is it clear? So it's the same thing here. If they have not given you the process of DNA replication, but they've brought a diagram and that diagram is showing that DNA replication is taking place. Is it clear? Is the one which takes place, the one which you're gonna describe. So you have to first identify it and then you're able to describe it. Therefore, you'll be getting those marks. Sir, will you do genetics? Definitely. I will do so. So that's why I'm rushing for this other content so that you are able to do genetics. All right, I'm done with that. Then we are going to protein synthesis. Before I go to protein synthesis, they can ask you a question that give the difference between the nucleotide of DNA and the nucleotide of RNA. In this case, they are looking at the nucleotide of which the phosphate is the same, but the sugar and the nitrogenous bases are not the same. So here you say deoxyribose sugar for DNA and the ribose sugar for RNA, and then here you say thymine for DNA and you say uracil for RNA. That is they are not talking about the difference between DNA and RNA. They are talking about the nucleotide of DNA and the nucleotide of RNA. I think, guys, I'm clear. Let me know if the sound is 100% right. Yes, because we had some technical issues. Ne? Yeah, so if we are 100% right, then let's continue. All right. Here is protein synthesis. What do you need to know about protein synthesis? You need to know how to label what is happening in transcription and what is happening in translation. Yes. Transcription means what? Formation of messenger. They asked that question some time back and people were failing it. Translation means what? Formation of a polypeptide chain. Please don't confuse polypeptide chain with the DNA, with the protein. A polypeptide chain means less than 50 amino acid. If it is above 50 amino acid, we call it a protein. Is it clear? Yes. So now let's label. Let's label. What is W? W is what? Let's label together. However much you are not writing it, but before I say it, you are able to give me the answer. Ne? Like say it in your heart, or you can say it loudly where you are, where you are sitting, so that when I say the answer, at least you get it right. If you get it wrong, then it means that you need to know it better. All right, let's continue. What is W? W, DNA molecule. Yes. What is Y? What is Y? Y. Y is amino acid. What is Z? Z is the ribosome. And then what is X? X is the messenger RNA. Good. Uh-huh. What is this? Okay, now since this one is not labeled here, now you're gonna tell me from the comment section that what is this. I'm not going to say it. I will first see the answer there, then I will continue. What is this? What is O and what is P? Ne? So we have O and then we have P. We have three bases on DNA. We call them code. We have three bases on messenger. We call them codon. We have three bases here. We call them anticodon. Someone is saying polypeptide chain is O, and then polypeptide is saying peptide bond, and then okay, I've got you. Thank you so much. All right. Guys, O, thank you so much. O is polypeptide chain. Let me show you what is a polypeptide bond. Let me zoom this out. Let me show you what is a this is the polypeptide. This is a peptide bond. We don't have polypeptide bond. No, we have a peptide bond. You see these lines? These are the polypeptide bonds. This circle is an amino acid. So the whole structure is polypeptide chain. Is it clear? I think I'm clear there. Thank you. Thank you so much, guys, who gave me the answers. Yeah, at least it shows that you are learning something. All right. I think I'm done. So if they ask you to describe the process that takes place in the nucleus, then automatically you describe transcription in terms of synthesis. So this is the nuclear membrane, while these small holes are the nuclear cores. Yes. Hey, can we go? Yes. So you go to transcription, DNA double helix unwind. DNA double helix unwind. The double helix DNA unwind is the same thing. Weak hydrogen bonds break to form two separate. The word separate must be there. To form two, don't say to form separate strands. How many are they? To form two separate strands. Then we are saying that each strand is acting as a template when it is about DNA replication. But when it is about transcription, you say one strand is acting as a template. Using free floating DNA nucleotide if you're talking about DNA replication. But if you're talking about transcription, you are forming a messenger. Therefore, we use using free messenger RNA nucleotide. Is it clear? Yes. From the nucleoplasm. A messenger RNA which is complementary to DNA is formed. Is it clear? Whereby adenine goes pairs. Let me be clear here. It's better to say that adenine is complementary to, use the word complementary to. Why? Because here we talked about complementary. Adenine is complementary to uracil, and guanine or cytosine is complementary to guanine. Now, messenger RNA with coded information. This is a marking point. Whatever you see in red is a marking point. Coded information, yes, for protein synthesis, and then now it moves out of the nucleus through the nuclear pore to the ribosome in the cytoplasm. So basically, guys, you know this. So I don't need to repeat this over and over and over and over again. But I'll show you where you get sucked by this topic. I'm coming there. Sir, did he do meiosis? Not yet. Not yet. I'm going to go there. Yes. 20 minutes ago. What does that? Oh, all right, guys. Hey, guys, just start. Just start. Just as long as you are here, let's study together. All right. Each here you talk about. Now, you see this. That is a polypeptide chain. You see this A. That is a peptide bond. You see this. That is amino acid. You see this. That is transfer RNA with the amino acid. Transfer RNA. You see this. That is anticodon. You see this. That is amino acid. So we say that when the transfer RNA anticodon matches with the codon of messenger, this transfer RNA will bring this required amino acid to the ribosome because this structure is the ribosome. Is it clear? Yes. Now, once it brings it here, then the amino acids are joined together by the peptide bond to form a required protein. That's it. So, guys, I think you got it right. So that is it. So each transfer RNA carries amino acid. When the amino anticodon of the transfer RNA matches the codon of the messenger, the transfer RNA brings the required amino acid to the ribosome. Then we are saying that now amino acids are attached by peptide bond to form a required protein. Simple. Guys, don't make a mistake to go when you don't know how to label this. You can't. You can't. Mutations on DNA. Yeah, we're gonna talk about it. But before we talk about it, answer for me this. Let's answer them together. All right. I'm not answer for me what is X and answer for me what is Y. I want to know what is X and what is Y. So as you're answering, I want to give you something here. Uh-huh. Okay. We are starting. What is X, guys? Hey, I'm not seeing your answers. What is X? Okay. I'm not going to answer X. What is X and what is Z? What is X and what is Y? So Z is messenger RNA. Uh-huh. Then give the nitrogenous base sequence of DNA triplet complementary to middle codon. So now when we look at middle codon, now I've robbed it off. I don't see the answers. Where are you? Okay. X is the amino acid. Don't be scared to give me the answer. And then Y is the anticodons. You understand? Because they're looking at these three bases. So they are anticodon. Now you know the answer you are trying to give me. All right. So this is amino acid is anticodon. They're saying that give the nitrogenous base sequence of DNA based triplet complementary to the middle codon on the Z. On the Z, where is Z? Yes. So meaning the middle codon, meaning that they're looking for this, of which they're saying that the DNA trip, this is a messenger, so it's gonna be A goes with the T, and then U goes with the A, and then C. So that's how it's gonna be. Is it clear? Yeah. Then they're saying that describe name DNA base strip. Okay, we're done with that. Describe name and describe the process that occurs in the diagram above. So here we are talking about translation. So you describe translation. I think I'm done with that. Let's go to the most difficult part. Imitation. Imitation. All right. We have two kinds of imitation. There is a mutation which results in a change and a mutation which doesn't result in a change. And how do we ask them in the question? Then there is another question which we ask about this mutation. Guys, DNA profiling, please don't go without knowing it because I have not gone through it. I expected it to be, you know it. All right. What will happen if a mutation changes the base triplet one, yes, from GT from ATG to ATA. Guys, look critically. You see, even I've reduced the voice so that you can understand it better. Ne? Look critically. I repeat. All right. Thank you so much for those who participated. Now, we are saying that uh, when they give you this information, first check what is here and what is here. Oh, I forgot to talk about the other thing. What is it? Yeah, because it's going to help me to answer that. Codon, these are three bases found on the DNA. Code, these are three bases found on DNA. Codon, these are three bases found on messenger RNA. Anticodons, these are three bases found on the transfer RNA. So, how many amino acids? Yes, are in 20 are in 20 anticodon. Remember, each anticodon, look at it here. Each anticodon, because we say that Y is anticodon, eh? Each anticodon codes for one amino acid. If they have 20 amino acids, automatically there will be 20 anticodons. But if I have 20 amino acids, how many bases will be there? It will be 20 times 3 because each anticodon has 3 bases. So if I have 20 amino acids, how many anticodons will I have? It will be 20. But how many bases will I have? It will be 20 times 3. Why 20? Because each anticodon codes for the same amino acid. One anticodon is equal to one amino acid. 20 amino acids are equal to 20 anticodons. But each anticodon is equal to 3 bases. So if I have 20 amino acids, it means that I have 20 anticodons. Each anticodon is 3, so times 3, which gives me 60 bases. Is it clear? All right. Sometimes we bring these questions where they ask you this and you go off. Now, since I know that, now we come back here. I said that when they give you this table, first identify what is here and what is here. What is here because of the T, it means that this is DNA. However much, they have mentioned it here. So you have DNA, but here you have codons, meaning that you can't read it from here and you bring it here. What you need to do is you have to convert this and then you read it there. So if you do that, yes, if you do that, then it means that now this you will be able to convert. For example, what they give us is ATG. So ATG. Sometimes when you go direct, you'll find the answer there, but that answer will be wrong. You understand? You have to convert it into ATG is gonna be A goes with U, and then T goes with A, G goes with C. So you look for this here. UAC, UAC is tyrosine. Oh, even I pointed it direct, is tyrosine. Now you come here. You look at this, the one which has changed to ATA. You convert it into UAU. UAU is what? UAU against tyrosine. So meaning that this, thank you. This is tyrosine, and also this is tyrosine. So it means that there will be no change in the protein, however much there is a change in the sequence of DNA. So how do you write an exam? You tell us that this changed to this and this changed to this, of which these two, they code for the same amino acid, which is the tyrosine. You have to mention this and you have to mention this. This changed to this and this 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 changed to this. You understand? And this codes for tyrosine, and this codes for what? For tyrosine too, which does not change, the protein. Why? Because it is the same amino acid. What about when there is a change? What about when there is a change? Okay, right. What about when I have T, and this TTA changed to TTT, of which TTA, this one is gonna be AAU, it's gonna be U, it's gonna be AAA. Yes, there is no mutation, there is no change. Boy B, there was a mutation, but this mutation had no effect because the mutation is not the change in the protein. No, the mutation is the change in the base sequence. It's called gene mutation. So if the base sequence have changed, then automatically the automatically this guy confusing me. Ah, who is confusing me? Okay, automatically there was a mutation, because a mutation means change in the sequence of DNA bases, or the change of the structure of chromosome if it is chromosomal mutation. But if it is change in the sequence or the bases, then we call it gene mutation. So if this mutation, if the change leads to the same amino acid, therefore they won't be effect on the protein. But if the change in the base sequence leads to a different amino acid, then there will be a change in the protein. Is it clear? I told you this is you call it a devil. I don't know why you call it a devil. Yeah. So now, base triplet number 4 changed this one. Changed from this, and then it went, meaning that this A was replaced by T. Cool. So when I change this to this, why do I need to change it? Because I can't read this direct here. Why? Because this is codon. So I have to change this DNA into anticodon. Sorry, into codon. So now AAU is what? AAU is valine, isn't it? And then now TTT, when I convert it into messenger, is going to be AAA, which is AAA. AAA is proline. You see that a different amino acid is formed. So to get those 3 marks, you have to tell us that Yes, this changed to this. Sorry. Yes, this changed to this, of which this code is for valine and this code is for proline. Therefore, different amino acids will be coded for, and then a different protein will be formed. So, in other words, let me give it a summary. When you convert this and you see that it's the same amino acid, then you tell us those amino acids, ne? Then you tell us that there is no effect on the protein. But if you convert them and you see that the first one codes for one amino acid and the second one codes for the second amino acid or another amino acid, then it means that there will be an effect on the protein. Is it clear? So please don't go the paper without knowing this. However, there is sometimes this question they ask you. They say that what will happen if the base sequence of DNA is changed? What will happen to the protein if the base sequence of DNA is changed? So that question is open. So you tell us that change in the DNA sequence will result in a change in the sequence of messenger RNA, which will result in a different sequence of transfer RNA bringing amino acids, isn't it? Then which results a different protein. Or if they ask you from messenger RNA, then you start from messenger. A change in the sequence of bases of messenger will result in a different sequence of transfer RNA bringing amino acid, and then it will result in a different protein. Yeah. I think I'm done with that. Please don't go without knowing that. Lastly, when they bring here, they say it's DNA, and here they put anticodon. What do I do? You change this DNA into messenger, and then you change the messenger into transfer RNA because you don't have anticodons. If this is anticodon, because you don't have anticodon on messenger, so it means that you convert it 2 times. Yes, you convert it 2 times so that you're able to read what is here in the table. Please assure me that we have the 27 marks now. And you know our method, that by the time we are done, we will be done with the reading only to practice, and you pass the paper by smashing it. Meiosis. If you have any challenge, let me know. Meiosis, you know the functions, why is meiosis needed? Basically, meiosis is needed for almost 4 reasons. Even if you know two, is better. Number one, to produce haploid gametes. Number two, genetic variation. It's better to say genetic variation. Don't say variation. Because variation can be genetical, or it can be mm, mm, environmental. When you go, my girls, when you go to when you go to ShopRite, and then you buy gentle, you could eat gentle magic, you change your skin to be lighter, ne? That is a variation before and after. You understand? But that's not genetical, because the kid you produce will be like your original color. You understand? So that's why we hear when you say variation, it's better to say genetic variation. All right. It maintains the chromosome number. Those are functions of meiosis. The first one, haploid gametes. The second one, maintaining the chromosome number from generation to generation. The third one, you can talk about yeah, you said formation of haploid gametes, ne? Yeah, genetic variation, haploid gametes, and then maintain the chromosome number from generation to generation. All right, let's continue. Where does meiosis occur in animals? In animals, ovary, to produce ovum. That's female. Testis, eh, testis. This is one, this is many. So testis are in male to produce spermatozoa. Cool. In plants, ovary produce ovules, anthers to produce pollen grains. Think I'm clear there. Mm. You confuse yourself by unreplicated chromosomes and chromatids. Chromatids, we only call them chromatids if the chromosome, these are linked together. If they are separated, they are not chromatids. We call them unreplicated chromosome before DNA replication. Unreplicated chromosome. You see? Unreplicated chromosome. But once DNA replication occurs, they become like this. That's why you call them replicated chromosomes. Replicated chromosomes. Well, here we call them unreplicated chromosomes. You think I'm clear there? Huh? We continue to the next question, or karyotype. When you talk about a karyo means nucleus. Which type of nucleus do you have? Karyotype. So that's the meaning. So then when they ask you to describe the structure of a karyotype, it can come in 6 marks. Describe the structure of a karyotype, 6 marks. This one means this one means that you tell us the arrangement of chromosomes. Karyotype, you are telling us arrangement of the chromosomes. How many are they? What are they in a cell? So you arrange the chromosome from pair number 1 to pair number 22. We call them autosomes. 23rd pair, we call gonosome. 23rd pair has X and Y. If it's X and X is a male, is a female. If X and Y is a male, and Y is shorter than X, or X is longer than Y. So you just describe what is happening there. Is it clear? But when they ask you how many chromosomes do you see there, then here you have to look at it. Don't rush, because rushing will never bring anything good. Sometimes we make this one 2 or 3, like that, and then ask you how many autosomes are there. You know that autosomes is from 1 until 22, isn't it? Then we ask you how many autosomes are there. But there is another one here, because when you know that it's 21 pairs, therefore is 40, 42. Did I say 21 pairs? It's 22 pairs. It's 44. Then I say, yeah, I know it. Can't there is something there? They have put another one there. Oh, this one is not there. You see? So please count them. Count them. And sometimes they bring it when it is not about a human karyotype. It could be something else. So please, please, please follow based on what is given. All right. Karyotype. When it comes to these phases, in prophase, you need to know what is crossing over. Please, don't go to the paper without knowing crossing over, unless you are burying yourself when you are still alive. The word homologous chromosomes aligning together is a key point. Is it clear? Then the word chromatids of these homologous chromosomes. One chromatid from one homologous chromosome, or each homologous chromosome overlap is a marking point. But where do they overlap from? At a point called chiasma. Is it clear? Is a marking point. At a point called chiasma. So we give you another mark there. Then the chromosomes will have 2 new combinations, meaning that each from these homologous what? Chromosome. Meaning that this one will have one part of this and this one will have one part of this. That is the function of crossing over. Basically, these chromosomes are not genetically identical. No. They are this, they call them homologous. They are the same, but they are not identical. So does it mean that because you're a Zulu and your father is a Zulu or your mother is a Zulu, does it make you to be identical? No. So because they are the same family, doesn't mean make them to be like identical. They have the same location of genes, but they don't have the same genes. Like here, you see, here it is capital letters, while here is small letters, but it's the same type of gene. That's the meaning. Mm. So when the crossing over takes place on top and they ask you to draw, when crossing over asks you like on top, so the variation must be on top. When it's starting from here to there, it must also show that. So don't just draw because you want to draw or you want to finish. You'll get a 0 with big eyes. I'm telling you. So I think I'm done with that. Let me know if you have a challenge, ne? Yeah, because I'm here to smash the challenges. Guys, what happens in metaphase 1? Metaphase 1, homologous chromosome line up at the equator randomly in 2 rows. Randomly in 2 rows. I think I'm clear there. I don't want to emphasize. It must be checked randomly. We don't know whether this chromosome is going to be here or is going to be there. Or this one is going to be this side. And the moment they swap, those are different chromosomes. Is it clear? Yes. So they align along the equator in homologous pairs. Is it clear? And then in anaphase, the homologous chromosome, they are pulled to opposite poles, meaning that they separate. Yes? Pulled to opposite poles, meaning that they're what? They are separated. So don't go to the paper without knowing. Process of crossing over, metaphase, anaphase. You can fail to know others, but you can't fail to know those ones. Cytokinesis. Cytokinesis division. Cyto means cytoplasm, kinesis. Basically using this so-called energy. So the division of the nucleus, we call it cytokinesis. Hey, did I say nucleus? Division of the cytoplasm. So please, we talked about this, but you need to tell me before I go there. What is non-disjunction? What is non-disjunction? Failure of the chromosomes to separate during metaphase is what called non-disjunction. And this non-disjunction is going to lead to Down's syndrome. Down's syndrome. So we call it non-disjunction, and then it's going to lead to Down's syndrome. I think I'm clear there. When they ask you the difference between meiosis 1 and meiosis 2, you don't need to suffer. Just go to meiosis H phase. Then you tell us. In prophase 1, we have crossing over. In prophase 2, we have no crossing over. In metaphase 1, homologous chromosomes align along the equator. In meiosis 2, individual chromosomes align along the equator. You see? In meiosis 1, under anaphase, chromosomes, homologous chromosome moved opposite poles. Under the other one, we say chromatids move to opposite poles. Is it clear? In meiosis one, you result in in two daughter cells. In meiosis two, you result in four daughter cells. So you see, we have a lot. You can go phase by phase, phase by phase. Someone is asking me, wait, isn't it anaphase one? What is it? What is anaphase one? What do you mean by anaphase one? I don't know what you are saying. Anaphase one. Random, you mean non-disjunction. Non-disjunction can occur, difference, sorry sir, what are the different, okay, I'm going to talk about it. Non-disjunction occur in anaphase one and anaphase two. In anaphase one and anaphase two. Yes. And sometimes they bring this cell and then they ask you that you explain what is happening based on this anaphase one. Is it for anaphase one or it is for anaphase two? In the difference between meiosis, what are some of the similarities? Similarities between meiosis and mitosis, both they require the nuclear, meaning that there is nuclear karyokinesis, there is cytokinesis. You see? Yes. Both involves the same phases. Interphase, no, prophase, metaphase, anaphase, telophase. Both there is involvement of DNA replication. So that's it. And then the difference, actually, meiosis too looks like mitosis. You understand? So basically, in meiosis, there is variation. In mitosis, there is no variation. In meiosis, you result in two daughter cells. In mitosis, you result in two daughter cells. In meiosis, you result in four daughter cells. In meiosis, you talk about having half the chromosome number. In mitosis, you don't have the chromosome, meaning that the chromosome number are what? Are maintained. So, guys, you need to know this. There is no crossing over, there is crossing over. It is easy. Guys, can I tell you something before we go to genetics? You need to read loudly if it is possible. When you are reading, make sure that you read loudly if it is possible. Make sure you read loudly. Yeah. So we are going to genetics, ne? But before we go to genetics, let's just do a few questions here. Just one, two, and then yeah. All right. Shabalala, what are you saying? All right. The diagram below represents the chromosome of human somatic cell of two individuals. I found out that people confuse what is called orosomes and gonosomes and somatic cells. Guys, when you talk about orosomes, orosomes, these someone saying restart the live. Okay, guys, can you hear me? Let me know if you can hear me. Ne? Because I think there is some technical issues. It was, I think, some technical issue with the internet, but I think now they have resolved it. Ne? Thank you so much for making me know that now you can hear me, and thank you so much, guys. Those who told me that you can't hear me. Ne? Yeah, thank you so much. All right, now let's continue back. Let's go back to our discussion. Let's go back to our discussion. So I was saying that. Now I'm lost. I don't know where I stopped with you. Okay, let me just do like this so that maybe we are able to be at the same page. All right, I'm saying that here is a question. Ne? These diagrams Yeah, I was saying that some people, they confuse what you call the somatic cell, the gonosomes, and the autosomes. Autosomes, they go with the somatic cell. These somatic cells are cells, are body cells. You understand? They are body cells, meaning that these cells, they have the chromosomes inside. So when they say somatic cells, these are body cells. Cool. Name the specific type of chromosomes numbered 1 to 22. You see, how do you call this up to there? So we call them autosomes. Each of the pair shown in the is a homologous pair of chromosomes. State the origin of the chromosomes in the homologous pair during the zygote. Obviously, um, um, one will come from the mother and one will come from the father. What are stem cells? We will talk about them, because that is genetics, and genetics is our next topic. We're just finalizing with the meiosis. And then they're saying that, uh, what is it? State the origin. One chromosome comes from the father, and one chromosome comes from the mother. Now, here is another question. List three characteristics chromosomes in a homologous pair have in common. If these are homologous chromosomes, what do they have in a pair? We saw it when we were discussing about, yes, one from the father, one from the mother. We saw it. We say that, first of all, they have the same size. Ne? They have the same shape, same size. If you look at them, same size. Ne? They have the same shape. Ne? They have the same locus or loci for the genes. They have the same size, same height, same shape, yes, and then same location of, not same location of chromosomes. No, not the same location of chromosomes, same location of genes on the chromosome. You understand? Because it's already a chromosome. Thank you so much. Thank you. All right. Explain one observable reason. Explain one observable reason why the two individuals are not identical twins. So, what do you say when it comes to that? So, the observable reason you can give here is they are not identical. Why? Because this is a female, this is a male. So, these are two different chromosomes. Sorry, individuals. You cannot have identical twins who are different sex. You understand? You cannot have identical chromosomes which are different sex. So it cannot happen that way. So this one, how many marks? Different sex, whereby individual one is a female, is a male, while individual two is a female, because of this same size of shape, same size of chromosome, and then here different sizes. Yes, thank you so much. We got the next question. Do you have any question? Okay, that is it. Let's go to genetics. There are some things I need to emphasize here. Terminologies, you need to do that. When it comes to the format of writing a genetic cross, you not write phenotype, genotype, meiosis, fertilization. They need to be in the corresponding position. Is it clear? Oh, before I go there, ish, just need to tell you this before I go to genetics. Guys, when it comes to meiosis, when they say that, how many chromosomes do you see? How many chromosomes are there in that cell? And this cell is showing you this. Let me just show you before I go back. You see these chromosomes, and then they ask you how many chromosomes are there in that cell. Don't say 46 chromosomes because this is not human cell. If they don't say human cell, please just say 1, 2, 3, 4 chromosomes. How many will there be at the end of meiosis? Because these are in pairs, therefore there will be 2 in each. How many chromosomes will there be in the same cell of human under the same phase? That's when, because this is meiosis 1, meaning that metaphase 1, yes, then you talk about 46 chromosomes because they mentioned about human. But if they don't talk about human cell, please do not do that. Do not do that. They can ask you also, how many chromatids are found in this cell? So each chromosome has 2 chromatids. So it's 1, 2, 3, 4, 5, 6, 7, 8. So they're going to be 8 chromatids. Simple. Let's go back. I think that's what I wanted to talk about. Yes. So when it comes to this genetic cross, I say that please make sure that you write phenotype, genotype, meiosis, fertilization. You know why? Because we give a mark here, we combine this and this, we give you a mark, and then we combine this and this, we give a mark. So it is P1, F1, we give a mark. Meiosis and fertilization, we give another mark. So you go to the paper we have when you have 2 marks. I wonder those people who get 0. Even when you can't do this, no. If you see genetic cross, yes, P1, F1. If that is P1, F1. If it is second generation, then you say P2, F2. Is it clear? So phenotype, you get it from the question. They will say that Karabo who is anemic married Joseph who is normal. So you say that Karabo anemic, Joseph normal. Crossed, you see, you understand. Then genotype, genotype, that's where the problem comes in. You need to find out whether it is homozygous or is heterozygous. If it is homozygous, then is it homozygous dominant or is it homozygous recessive? Uh, Someone saying that, am I still talking? Yes. Yes. Do you have any challenge there? Yeah. So I think I'm clear there. So that's it. Now, when it comes to this, when you're marking, yes, we shall mark, we shall give you a tick here, we shall give you a tick here, we shall give you a tick here. Remember, this is from the question. So you have 3 marks, mahala. You understand? You have 3 marks, mahala. So now, since you have 3 marks, mahala, so now you have to find this one correct from the question, whether it is dominant or recessive. Once you get that, now, for example, let me show you another mistake students do here. You have capital T, small t, capital T, small t, for example. Then meiosis takes place. This one must be separated. This is capital T, this is small t. This is capital T, this is small t. Guys, please, I beg you, don't write this one here and this one there. We shall call this one, how are we gonna call it? We're gonna call it a genotype. So there must be space in between. That's why I prefer to tell my kids that, you know what you guys do, what you should do. Number 1, just capital T, put a comma, small t. Even if they're together like this, at least we shall know that there is a comma there. Or don't make them to be like the beach. You know, at the beach, what they do, people are naked. They only have, they are covering a specific part. So don't leave them like that. Just dress them up, put them on in circles, dress them up. Ne? Yes. Once you dress them up, even if they are too close, still we shall give you a mark because you have circled them, you have separated them. I think I'm clear there. Then, right fertilization, now start crossing. If you don't cross like this, use a Punnett square. You understand? So the Punnett square, bring this here, and then you say capital T, small t. Then you bring this here, capital T, small t. So we shall give you a mark there, we shall give you a mark there, we shall give you a mark there. Then you cross capital T, capital T, capital T, small t, capital T, small t, small t, small t. We give you another mark there. Are you seeing? Then you write the genotype, the ratio, the phenotype, the ratio, depending on what they have asked you. But I prefer write everything because whether you have written it or not, we won't cross you. But if you have not written it and they wanted it, you lose a mark. So just write everything you know about it. That is the first point. So the second point, write everything. Why am I saying that? When you say P1, don't wait to write F1. Start with the P1, F1, because sometimes you write P1 and you forget F1. Please, 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 please. Eh? Yes. All right. Another thing which you do, you guys, when you are doing this genetic cross and makes you lose the marks is, now you might see your teacher is a little bit sympathetic to you, but when it comes to the marking, they will not be sympathetic to you because they won't know you, and no one will be willing to lose, let me say, the job they have signed for. So, meaning that please do things which are right. So, for example, you write here capital T, small t, capital T, small t, and then you start crossing. You know what you cross? Let me just write it here. Capital T, small t, capital T, small t. Then you cross like this. Capital T, small t. You cross like this. Capital T, small t. You cross. You cross like this. See? Capital T, small t. Cross. There's a way how you cross. You cross like this. Eh, there's a way how you cross. You cross like this. Then you say capital small t, small t. So we ask you, what is this? What is that thing? Where is this? Is it this or this? Where is this one? So I prefer, if you don't know, it's better use a Punnett square method. So it's better that way. Is it clear? Yes. Lastly, about this. When they say that P1 F1 is a compulsory mark, né? I'm just giving an example. And the meiosis and fertilization is another compulsory mark. Two compulsory mark. And then they've given you 6 marks. Né? In most cases, the genetic cross is 6 marks. Né? So what they do is they first subtract the compulsory mark and they give you any other 4. If you don't get this right and don't get this right, you get 6 here, they will give you 4 out of 6 because you have failed to score a compulsory. So that's why I'm saying that write everything. How fail? Write everything. Fail. Ne? Yeah. Then once you do that, you'll be able to succeed. All right. Let's continue. Uh-huh. Dominances. When you talk about complete dominancy, incomplete dominancy, codominancy, here we are talking about Mendel's law of dominance. In Mendel's law of dominance, when you are talking about Mendel's law of dominance, it means that one allele is completely dominant over the other in a heterozygous state. When you have a capital letter, small letter cannot speak. You understand? If there our president Ramaphosa is the president, if it is if he is available, you cannot, the vice president, Natural selection is very challenging. Prepare more questions. All right, Karabo Karo. All right. The vice president cannot speak if the president is available. That's the meaning. So when a dominant allele is present, a recessive allele cannot what? Cannot do anything. Is it clear? Yes. Sorry. So this is not my concern. My concern is coming. It's about the pedigrees. I know these ones you can kill them, but when it comes to pedigrees, sometimes you mess up, and you know yourself. All right, we will sort it out. So, someone asked me about that question. So now, look, when this is complete dominance. You have a red, you cross to the white, you produce a red. You produce one of the colors, then it is complete dominance. If they bring such a question, please know. All right. Thank you so much. I hope now you can hear me. Today, I think I've been with the problem with the network. Right? Network has been a very big problem. Now. All right. I hope now we are together. Can we continue? I hope you are hearing me now properly. All right. And we continue to the next question. We continue the next question. Then when you cross red with white and you produce an intermediate that is incomplete dominance. One of the allele, neither of the allele is completely dominant over the other. And in this case, we use capital letters. Capital R for red, capital W for white. When you cross the two, you produce capital R, capital W, which means pink. That is incomplete dominancy. When you produce a mixture, there is no difference between incomplete dominancy and codominancy when it goes to the crossing. It is the same. Is it clear? It is the same. Still, it's going to be capital R, capital, for example, W, and then you produce capital R, capital W. So it's the same style. But the only thing which helps us to know is the phenotype. When you produce a mixture of the two colors, then it becomes what? It becomes codominance, which means codominance means together. The two colors are existing at the same time. Are we together? Rivonia, are we together? All right. Rivonia, you let me know, eh? Yeah, if you have a challenge. Sex determination. Here, when you talk about sex determination, you just describe basically a karyotype. Nothing else. 22 pairs of chromosome in the female orosome. From 1 to 22 is orosome. The 23rd pair is gonosome. When one when X matches with when X of the female of the ovum fertilized fertilized by the X of the sperm, you produce X and X, which is a female. When the X of the ovum is fertilized by Y of the sperm, it becomes X and Y, which is a male. We have 50% chance of having kids who are female and 50% chance of kids who are male. So that is sex determination. They can ask you to describe it in words or describe it in terms of what? In terms of genes or genetic cross. Yeah. Aha. Pedigree is starting now. Pedigree is starting. Call your sisters, your brothers, your uncles, your what, whoever is having a challenge here. Please take note. If you have not taken a drink, take it. Relax. No one must disturb you. We start the pedigree. Okay. sex determination. Sex determination means sex-linked genes. These are genes which are carried on X chromosome or sex chromosome. You understand? Meaning that you only find it in that sex. So if it is the disorder, then it means that these are disorder caused by genes which are carried on X chromosomes. So examples is hemophilia, color blindness. Night blindness is also an example, but is not examinable. You understand? Yes, but I don't think if they ask you and you write it, uh, they will cross it. Anyway, it's better to write what is being asked. All right. Should I start with a degree first? Okay, let me just highlight this. I want answers now. How many genes control blood group? Write for me how many genes control blood group. Ibafetu, write for me how many genes control blood group. And then, yeah, I continue. So, yes, how many genes control blood group? Let me know, let me know, let me know. Let me know as I'm preparing for you here questions. Yes, this question will work. Okay. I don't see the answer. 3. Okay, thank you so much. Any other answer or the same answer? Yeah. Sorry, 1. Hey, you are giving answer and then you are crossing. Hey, people are saying, yeah, yeah. Hey, people, you must tell me. Ne? Okay, 3, 1. All right. How many genes control blood group? How many genes control blood group? Hey, why writing? Guys, it is one gene. One gene control blood group. Thank you so much, guys. Which gene is that? Gene for blood group. Hey, do you have any other gene? It's gene for blood group. Ne? How many alleles? There are 3 alleles. That's why it's called multiple allele. 3 alleles. Allele for A, allele for B, and allele for O. And when you are writing, don't write it like this. We will cross it. You'll get a 0. Don't write it like this. O. And then you say like that. You'll get a 0. However much some books, they allow it in other books, not if you look at the internet and the other books, they allow it, but it's wrong. When it comes here, we will cross it. So, how many alleles? 3. How many genes? 1. How many genotypes? There are 6. 6 of them. And how many phenotypes? There are 4 of them. Is it clear? Yes. Then, is it possible to have, I want yes or no. Is it possible to have, I hope those people who have gone for drinking water, you are back because I want to start the pedigree. Is it possible to have blood group a man of blood group A marries a lady of blood group B and they produce a kid of blood group O? Is it possible? Tell me answer yes or no. A man of blood group A, someone who is blood group A, marries someone who is blood group B, and then they produce a kid of blood group O. Is it possible? Is it possible? The answer yes or no. Just want to see yes or no, yes or no, yes or no, yes or no. Yes. Tell me what you know so that yes or no, yes or no, yes or no. All right. Yes or no, yes or no. Thank you, thank you, thank you. All right, guys, the answer is yes. It is possible. It is it is possible. Why? Look here. Look here on our distinction material. If you have blood group A and you are heterozygous, and blood group B, you are heterozygous, you can produce blood group O, whereby this small allele combined with this small allele, which gives me blood group O. You see? So it is possible for a person who is blood group and the person of blood group B to produce a kid who of blood group O. That's why we say that it is not conclusive. You understand? It is not conclusive to use blood group to identify the paternity or the maternity of an individual. It is not conclusive because many people have the same type of blood. Is it clear? Yes, it is not conclusive. We have, you will find out that your neighbor have blood group A and your what you have blood group A. So does this mean that your neighbor is your father or is your mother? No. What if you resemble the neighbor? I don't know. It's just you are resembling. Hey, they have never told you that when you resemble someone somewhere far. What if that person was your neighbor? Does it mean that something happened? Hey guys, I'm not in there. I'm just saying, eh? Yeah. All right. Hey. Uh-huh. Hey. All right, let's start. We are saying that uh if they ask you that it is, you just tell them that it is not what? It is not conclusive to use blood group as uh, as a way of identifying a paternity of an individual. So these are the questions we talked about them. Yes, cool. Mm, okay, before I go there, let me ask you. Okay, it's fine. I'll ask you the same time. So when it comes to dihybrid, guys, don't stress too much. Ne? When they ask you dihybrid, how do they ask you? Identify the alleles. No one is going to ask you to draw the whole thing. No. Identify the alleles, identify the genotypes, identify the phenotypes, identify, complete the table, and then identify the gametes. So they give you the gametes to identify the genotype, or they give you the genotype to identify the gametes. Is it clear? For example, something like this. If I have T, small t, capital R, small r, and ask you what are the gametes there. Obviously, I'm gonna say this is gonna go with this, so I'm gonna say capital T, small capital T, capital R. That's the first gamete. Now this one with this, gonna be capital T, small r. You circle them, ne? Then this with this is gonna be small t, capital R. Then this with this, small t, small r. You understand? Don't forget that Mendel said that the presence of one allele cannot influence the presence of another allele in a different location. That's why we start with a capital letter. But if they are different alleles, then automatically you keep the order. This is capital T, capital T, capital T, small r. But this one is small t, capital R, because T is different from R. You understand? Yes. T is T, depending on which allele is it determining. Which character is it determining? And then R is not T. Therefore, that's why we keep the same this order of starting with the T. But if it was capital R, small r, you can't write it like this. Capital small r, capital R. No, because it is the same kind of gene. Therefore, you have to say capital R, small r. Is it clear? Yes. Hmm. So, mm, no one is gonna ask you about those those what? Those those. So if they are like this, if it is, they can ask you the gametes from there. They can ask you the genotype there. They can ask you the percentage. If they ask you the percentage of those which are one seed, and then wrinkled. In this case, you have to add everything here, and then when you add everything you get a total. For example, 300. I'm not saying that it's 300, né? So you say that 100 and then divide by 300 times 100, so that you're able to get the percentage. You see, then you come here 290 divided 300 times 100, so you get the percentage. So you understand? So if they ask you the percentage, please don't confuse the ratio with the probability. If they want a ratio, it must be a ratio. They want probability, yes, you can say the probability, or the percentage. Yes, you can do the what? The percentage. Let's go to the pedigree, guys. Oh my God. My students, listen carefully. We have 4 different pedigrees. Finally, 4 different pedigrees. The first pedigree is the first pedigree is orozomo. Let me make it clear for you. The first pedigree is autosomal character, meaning that is carried on autosome chromosome, meaning that non-sex linked. I think now when I say it like that, it's clear. The second one is sex linked. You need to know that. The second category, so this is 1, this is 2. The second category is recessive allele or dominant allele. So is it caused by a dominant allele or is caused by a recessive allele? Is it clear? So now here, these two, this is what it comes. If they say some or same, married, carabo, this is a boy, this is a girl or a woman, whatever. However much this is a boy, you see is a boy, and this is a girl. And they didn't say that this is caused by sex linked. Please don't use X and Y. Don't. Use the letters they have given you. For example, in this case, first identify if it is caused by dominant allele or is caused by recessive allele. If it is caused by dominant allele, I will say that capital D, capital D. Whoever has capital D or capital D, small d, is caused by dominant allele. Meaning that whatever I have, if I have capital D, it will be that individual who is what? Shaded. Is a male, you see, but they have 2 letters because it is not sex linked. The female who, okay, the one which will be normal will have 2 recessive allele because the moment you have a dominant allele, that individual is sick. Is it clear? Now, if it is caused by a recessive allele, the one which is shaded, ne? Sorry. Someone is asking me, do you teach physical science? Yes. Yes, I teach physical science. All right. Now, the one which is shaded means if it is caused by recessive allele. If it's like this or it's like this, it means that these individuals are normal. You understand? Here it's caused by dominant allele. So these individuals are sick. So the one which is dominant, the one which are sick will be normal for recessive. And the one which are normal will be sick for dominant. So this one will be normal. And then this one will be affected. It is caused by recessive allele. All these ones will be affected. And then this one will be normal. It's caused by dominant allele. Hey guys, do you understand it? Before I go to the real example. So this is non-sex linked. If it's non-sex linked, whenever you say capital letter, that individual is sick or is affected. if it is dominant allele, caused by dominant allele. If it is a recessive allele, the one which is shaded, the one which is capital letter, it is normal. The sick one or affected one will be recessive allele. When it comes to sex linked, ne, dominant allele caused by dominant allele, the female will be that female will be sick, and that male will be sick. Is caused by dominant allele. And then this one is dominant allele because I have a capital letter somewhere. And then this one will be sick. Will be sick. Will be sick. You see, when it's caused by recessive allele, it is only one option for the person who is sick, meaning that the chances are fewer for this individual to be sick if it's caused by recessive allele. Let me go to the examples. I think I show you, you'll understand it better. Okay, here is the saying that this disorder, whatever, don't care, yes, is caused by an allele. You see, this disorder damages the arteries supplying blood to the brain. The pedigree diagram shows the inheritance of this disorder in the family. Did they mention that this is sex linked? No, they didn't. Since they didn't, and is caused by a dominant allele, so what does it mean? It means that whoever is affected has a capital letter R. That's the meaning. Whoever is affected has a capital letter what? R. You see? This is not sex linked capital letter. You see? You see? This individual is sick. You see? These individuals are what? Sick. And the one who is normal, you see, it has a small letter. Why? Because it is caused by a dominant allele. Bumelelo, are we getting there now? Yes. Meaning that this individual is affected and is a male. Because it's not is caused by a recessive, is caused by a dominant allele and non-sex linked, I can't use X and Y. Therefore, this one is normal, therefore is obvious. So it's gonna be small, small r, small r. The same way we said it here, the normal is this recessive allele. You see? And then this one will either be capital R, or capital R, small r. Let's find out which one is A. They produced a person who is small r is not affected. Automatically, this one will be this. One R came from him, one R came from her, so that they're able to produce this kid. Therefore, this individual is capital R, small r, so that they're able to produce small r, small r. It is caused by a dominant allele. So, shaded are dominant. Yes. In this case, because they say it is caused by a dominant allele, the shaded one, they are dominant. Is it clear, guys? The shaded one is caused by a dominant allele. Uh-oh. You guys? Okay, let me just finish up this, and then I have to break for some 10 minutes. Because I am a Muslim, I have to go for prayer. And I didn't see it, that it's already past time for my prayer, which we call Magharibi. All right. So we are saying that how many generations are represented in the diagram? So you count 1, 2, 3. Is it clear? Yes. So you count 1, 2, 3. Is it clear? Yes. So since you count them, so those are 3 generations. So it's cool. So boy tomorrow, obviously I'll pray for you. Then you're saying that yes, then we are saying that Give the letter of unaffected male. Unaffected male. So the unaffected male are those ones which is a box, you see, and is shaded. Give the letters of unaffected male, isn't it? Unaffected male is only H. Yes. So the answer is H. Because this disorder is caused by a dominant allele. So definitely it's only H. Give the genotype of A. I think we talked about it. Yes. Talked about it because this A produced a baby who is not affected. And remember, this disorder is caused by recessive allele. So it's caused by dominant allele. So if you are not affected, it means that you have small letter R, small letter R. Yes. Then, eh, sorry to disturb you, sir. Promote the name. What name is that now? Dumero. I don't understand. Okay. All right. You mean my name? My name is M. Saidi. Just call me M. Saidi. All right. Musa Saidi. All right. They're saying that individual B, G, individual. So we've said the genotype of individual A. We are done with that. Letters of individual not biological relative to individual A and B. When you come back here, individual A and B, the one which are not kids. Okay. This one, these two, they produce that and they produce that. You see? So this one just married this guy, and this guy just married this lady. So the answer is C and F. If this was like this, then automatically C would have been a child. But it's showing the marriage sign. Showing the what? The marriage, that this one they got married. So this A produced this and this one produced that. I think I'm clear. Yes, someone is saying, yes, how do we find is a sex linked? We're gonna come there. I have an example for that. So now that is the first scenario for dominant allele. So if it is a recessive allele, now this one is for a recessive allele. You see it? It is a recessive allele. If it is not a recessive allele, they will mention it still. So if you see it here, you see that you have a capital letter, have a small letter, but they didn't want to mention it there, but here they say that why is it a recessive allele? Is it clear? Yes. So you guys, let me leave you with this question. Then I go for prayer. Yeah. Mm, let me see if you can see it. You can flip your phone so that you're able to see it. I hope now you can see it. Yeah, 5 minutes. 10 minutes. 10 minutes. Yeah, you guys, please do for me those questions. Then I come back and then we smash it. But there is a key point there, Nana, who asked me, please, this shows that it's a sex linked. So meaning that, and sex linked, not just sex linked caused by a dominant allele. You understand? So we have seen the non-sex linked caused by a dominant allele. Now we want to see sex linked caused by a dominant allele. You understand? So please do that for me. Yes. uh I'll be back in very few minutes. All right. We are back. We are back. We are back. Uh, Someone is saying, okay. Okay, let me check and see. Let me check and see. Okay. Someone is saying, in the examination guideline, they didn't ask the difference between meiosis one and meiosis two. Do we still need to? Yes. Yes, please. Eh, can I tell you something? What else they didn't ask you and they asked it? Uh, Karyotype. Go check it. It's not there. They didn't talk about karyotype, but they ask it. Why? I don't know. So, you need to know it. You need to know it. All right, guys. Let's start. We saw that. Um, are we there, guys? Are we there? Are we there? Are we there? All right. They're saying that this disorder is a sex-linked genetic disorder. It is caused by a dominant allele. This one, ne? Meaning that whenever you see it is not shaded, it's shaded, it must have that. If it is a female, it must have at least one G. Same female, meaning that it's gonna have either that or it's gonna have that, depending on the offsprings they have produced. And if it is a male, it's going to have capital G, then it's going to have a small, it's going to have a Y. So that's what it means. So this disorder is a sex-linked genetic disorder. It is caused by a dominant allele, capital G. All right. So, guys, I think now you see that when they say that, Rivonia, what do you want from me? Smash, you want smash? All right, let's smash it. All right, we're saying that this disorder, ne? This disorder, they have said that is caused by sex linked. They will always say that in the question that it is caused by sex linked. If they don't say it's caused by sex linked, please do not use capital letter. Sorry, do not use X and Y. So that one is done. So number two, we have seen that whenever it is sex linked, whenever it's dominant allele, make sure that you use a capital letter. Whoever is shaded has a capital letter attached. Whoever is shaded has a capital letter is what? So if it is a male, obviously, then it's gonna be just a Y next to it. If it is a female, we're gonna have to look at the offsprings and we see whether this offsprings is there's the one who is affected or is not affected. Then we'll be able to know whether is he homozygous or it is heterozygous. Is it clear? Yes. So now let's find out on what they are saying. So it's better to write these genotypes and then you do the calculations you are doing. All right. They're saying that the diagram shows the inheritance in this family. Name the type of diagram shown above. Obviously, it's gonna be. I don't know why I like obviously. It was a student who said it, and I said stop using obvious. Now it is on me. Ish. It's not obvious. Yeah. So which type of diagram is it? It is the phylogenetic tree. Is it clear? Yes. How many females are in this family? How many females? Okay, let's find out how many females. Remember, female is a circle. Sometimes we bring how do they call it? Sometimes we bring the key, sometimes we don't bring a key. So whenever you see a circle, it is a what? It is a female. So it is one, it is two, it is three, it is four, it is five, it is six. There are six females. Is it clear? Yes. Mm-hmm. How many? How many females? How many males? in F1 generation. Listen, guys, here they're saying F1 generation. They are not asking in the first generation. No. F1 generation is different from the first generation. You are in F1 generation, but your parents are in first generation. I don't know whether I'm clear there. You, the kids, are in F1, the first filial generation. So, this is the first generation. This is the first filial generation. So, this is F1 generation. And these are the parents, they're in the. That's why you say that those are, daddy, those are your generation. Meaning that when and your daddy, you are in different generations. Is it clear? Yes. So they're saying how many in the F1 generation. Then we come back here and we look for that F1 generation. We have one and we have two. So there are two males in that regard. They're saying that Hobbies genotype. They want the hobbies genotype. What is hobby? Genotype. Obviously, this hobby is affected and is a female. So since it's affected, now we know at least we know where we are starting from. First of all, is affected. First of all, is a female. Is a female and is affected and is caused by a dominant allele. You see, it's caused by a dominant allele, meaning that this individual has a capital letter somewhere. But now we need to find out whether this capital letter, what are the kids of these two? We have a kid who is not affected at all. And this kid is a female, meaning that is having this. Remember, the one which has small letter are the one which are not affected. Therefore, how did she get this? One got from the father and one got from the mother, meaning that the mother is like that, is heterozygous for that condition. Yeah, is it clear, guys? Okay. Then they're saying that Anju and Pilusa have four children. Give the phenotype of their sons. Give the phenotype of their sons. They're saying Anju, and Pilusa have four children. So this is a male. Obviously, then we say that since it's a male, you write it there. Is capital is a male who is affected, meaning that capital G. And then this is the female, which is capital, but this individual is normal, meaning that he's having that. So meaning that they're saying the genotype of their sons is only talking about the phenotype of the sons. It's only talking about the sons. If the mother is normal, definitely all the sons will be normal. If one affected male, one affected male. Okay, yeah, thank you. Yeah, so meaning that here it means that mm, they are looking for the males. Remember, males are like this. So this got from the father from here, meaning that they're gonna get the from the mother. You see, so the genotype, the phenotype of the male is gonna be an affected male or unaffected, yes, unaffected male. So that will be the genotype. Give the genotype of their sons. Yes, it's unaffected males. Then they're saying that explain your answer. So it means that you explain them that since Anju is unaffected female and Pilusa is affected male, therefore the males will inherit one recessive allele from Anju and a Y chromosome from, yes, resulting them to be male, and the recessive allele will be for this. You see? Yes. So that is it. You see that if you practice these questions, it is easy. It is easy you are able to get the answers right. I wish you can pass and you have a beautiful life. Keep reading, you will pass. I know that. All right. So, guys, this is the first question. The second question on sex on dominant allele. So we say that if it is dominant allele, please, the one which is affected is the one which is having a capital letter. If it is a dominant allele and is not sex linked, it means that still the one who is affected is dominant, is capital letter. But even if they talked about the male, please do not use X and Y. You only use X and Y if they say that it is sexy linked. Then we go to this question. What about when it is non-sex linked recessive? It's caused by a recessive allele. Now, the one which is shaded will be recessive allele, will be small letters. So they are saying that it means that this one is affected. This one is affected. This one is not affected. So it means that this one is not affected. This one is affected. But now, what is the genotype for this? Because they produce this kid who is affected automatically, then it means that this kid is going to be small t, kid number 5, because the kid number 5 is affected, isn't it? So it means I got one recessive allele from there and one recessive allele from there, from the father, and one recessive allele from the mother. So when it is like that, then what does it mean? It means that this is normal, meaning that they have the one which is making them to be unaffected. Therefore, it's capital letter T and capital letter T. So automatically, these individuals are heterozygous, so that they're able to produce this individual who is homozygous recessive. Then they're saying that describe what is meant by autosomal disorder. It means that is a disorder whose genes are carried on autosomes. You understand? Yes, from pair number 1 to pair number 22. Yes, chromosomes. How many sons did the individual 1 and 2 have? Individual 1 and 2 have, they had one son. Why? Because this one is not a son. This is just someone who came to marry a beautiful girl. Number 4. Yes. Then they're saying that using individual 3, 4, and 7, explain why is it can be concluded that this disease is controlled by a recessive allele. Individual 3, 4, and 7. Yes, it can be concluded by that is because individual 3 and individual 4, they are normal. You understand? And they produced someone who is affected, meaning that they are heterozygous. This one is heterozygous, and also this one is heterozygous. So the recessive alleles combined and then produced individual who is affected. Meaning that, how do you say? You say that individual 3 and 4 are both normal. You understand? So they passed a recessive allele. Individual 3 passed, or individual 7 is affected with that disease. You understand? So those are 2 marks. So individual 7 obtain one recessive allele or small t from individual 4, and one recessive, say another mark, and one recessive allele from individual 3. You understand? Making the individual 7 to be affected. Simple. Will we get a mark if we say 3 and 4 are what? I don't see them. Okay, let me use this one. Will we get a mark if we say individual 3 and 4 are carriers? Individual 3 and 4 are carriers. Okay, let me check. Will you get a mark if they say individual 3 and 4 are carriers? Uh, You might, but it's better to say they are heterozygous. Yeah, it's better to say heterozygous. They are heterozygous. Mm. Ehen. So, guys, another example is a recessive allele but dominant, sorry, recessive allele but sexy linked. So, guys, we said that it can be dominant allele, non-sex linked, recessive allele, non-sex linked. Dominant allele, sex linked, dominant recessive allele, sex linked. You understand? So, you need to read. First of all, guys, make sure that you identify whether it's a recessive allele or it's a dominant allele. That's number one. Then identify whether it's sex linked or non-sex linked. Once you know that, you'll be able to answer questions which are concerning with this. Thank you so much. Huh. I think I'm winding up. I'm winding up with the genetics. So there are so many questions here. You can practice and practice and practice. Ne? Yes. What are stem cells? These are cells which have ability, uniqueness. They have ability, unique ability to develop and form other specialized cells. You understand? Have unique ability to form other specialized cells. Sources of stem cells, bone marrow. Yes, you need to know this one. At least you know two. Uses, they are basically used to replace the damaged cells. Or for, yes, damaged cells. So you have to know that. Cloning, guys, please. What you need to know about the. Did I talk about it? Did I remove it? Yes, it is here. Talk about it. Yes. About the genetic engineering. Whenever you talk about genetic engineering, guys, it must include the word manipulation. You manipulate. When you say engineering, you are manipulating the genes. Is it clear? Yes. So when you're engineering a building, you are manipulating the plan on the paper into a full structure. So, uh, advantages: expensive, interfering with the nature, and foreseeing long-term effects. Those ones, you know, guys. Ne? Yeah, you can just read them. Then now this one, cloning. When you have sheep A and sheep B, but you want to produce sheep 3, you understand? So what do? First of all, since I want sheep 3, this sheep 3 should resemble like sheep 1, then automatically what I need to do is I should use the genes of sheep 1. Yes, isn't it? So what do? I get a somatic cell, somatic, somatic cell. And then this somatic cell, I remove the nucleus and I keep the nucleus. I throw this away. Then I go to the ovum of this sheep. Why do I use the ovum of this sheep? I remove the ovum, I remove the nucleus, I throw it away. I remain with the nucleus. Sorry, the ovum structure, the outer structure, the membrane. Then after that, I combine the two with mild electric shock. And then I form a structure which looks like a zygote. Why do we say it looks like a zygote? Because we inserted this nucleus into the ovum, meaning the casing outside looks like the ovum. And because this cell is a somatic cell, therefore it's diploid. So it's like this thing is diploid and the casing is ovum. It's like when fertilization takes place, that's exactly how the ovum looks like. And then now I make this one to multiply by mitosis and then forms a ball of cells. And then now I can separate those cells and then I put them into different wombs to form embryos. And then it's able to produce this. How is the question being set here? The question is set in this regard or in this way. The first question is this. Why did we use the nucleus of this ship, not this ship? It's because we wanted the characteristics of ship 1, isn't it? Which method do we use to combine the two? We use the mild electric shock. Why did we keep the shell of chip number two? Because we obtained an ovum. Therefore, the resultant structure should look like an ovum. Huh. Which ship one or two will the offspring look like? It will look like ship one. Give a reason for your answer because we use the nucleus from ship one. You see? So you explain based on what is being. Then I can ask you other questions about biotechnology. Simple. Hmm. Welcome to evolution. Yes, I know that, guys. Evolution is a little bit simple. Why? Because you just recently did this. Someone saying finally. Yes, finally, we are there. We are winning. So, evolution here, I'm just going to, you need to know what is called a hypothesis. You need to know what is called a theory. A hypothesis is just a tentative um, um, statement which can be proven correct or wrong. It must be tested. Statement, a phenomena which is an inquiry which must be tested. Then a theory is an explanation of a phenomena which is accepted to be true and or is supported by facts, experiments, laws. It must is an explanation of something which has happened. Is it clear? Yes. So theory, I'm done. Evidences of this yes, variation. We talked about the causes of, did we talk about them? Yes, we talked about them. Cause of variation. Random arrangement of chromosome during metaphase, random fertilization, crossing over, and then mutation. But if they say meiosis, the cause of variation based on meiosis, then you talk about random arrangement of chromosome during metaphase and crossing over. Sometimes mutation is also there, but you write it at the end. So variation is the difference among organisms of the same species. Difference among organisms of the same species. That's why I always ask people, are you sure your father is your father? You don't ask that to your mom. Are you sure your father is your father? Yes, your father could be your father, but because of variation, because of crossing over which took place during the formation of these gametes, that's why you don't resemble him or you don't resemble your sisters. The only problem is that one I asked you before. What if you don't resemble him but you resemble your neighbor? All right. I'm just asking. Okay, they're saying that yes, it could be your father, but because of the variation, the genetic variation which occurred crossing over, then it made you to be different. Yes. So what's a species? You need to know that definition is a group of organisms consist of similar individuals capable of interbreeding. Capable of interbreeding. You don't stop there. You say produce fertile offspring. You guys, please help me. Don't use viable anymore because we have removed it to produce fertile offspring. A population is a group of organisms of the same species living together in a particular area. You understand? At the same time, that specific time, it should be living together, and they be able to interbreed. So that one you know. Aha, here is some small confusion. Types of variation: continuous variation and discontinuous variation. Anything which we are born with and it is exactly the same way you are born with, then it is follows under what you call discontinuous variation. Your blood type is discontinuous variation. If you are born blood group A, you are blood group A, you will die blood group A. You won't change. If you are born blood group B, you will die blood group B and your blood group B. You won't change. Is it clear? So whatever doesn't change from the time you are born until you die, then that is discontinuous variation. But the one which changes, for example, the size, the height, everything, we call it continuous variation. Yes, imagine. Yes, the size of your head now and the size of your head before. It's not the same. The shoe size, it's not the same. So those are continuous variation. So meaning that we have a range of characters. While here we don't have intermediates. Here we have intermediates. We have that dark skinned, light skinned, too light skinned, too dark skinned. So we'll find out the one which are in the moderate will be having high numbers. You understand? Yes. So here is blood group. Tongue rolling is also another example of discontinuous. If you know how to roll the tongue, unfortunately, I didn't put on the camera. Hmm. So theories, Lamarck's theory, Downey's theory. I can't repeat this because you know it from your, yes. But there is only few questions I'm going to ask you. Please, when they bring a question, make sure that you apply the way it's supposed to be applied. Ne? Yes. When they bring a question about snakes, use snakes. Don't use giraffes. You'll find people using giraffes when they're talking about snakes. Why is Lamarck's theory rejected? Lamarck's theory is rejected. That's what I'm going to focus on. Lamarck's theory is rejected because there is not enough evidence to show that the acquired characteristics are inherited. Don't just say that there is not enough evidence to support the theory. There is not enough evidence to show that acquired characteristics are inherited. Is it clear? So if they have given you an example, use that example. There is not enough evidence that the acquired characteristic of long necks are inherited. Do you see? The acquired characteristic of big shoe, big leg, big what are inherited. Number two, organisms don't evolve because they want to evolve. These giraffes, they didn't become longer because they wanted to become longer. They evolved because of natural selection. Is it clear? So please, 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 it's rejected because there is not enough evidence to show that the acquired characteristic of long necks you apply based on the question. Unless the question is just flat, then that's when you say there's not enough evidence to show that acquired characteristic is so. But if not, make sure that you give it right. Huh. So what is the difference between Downey's theory and Lamarck's theory? Lamarckism, when you look at it at first, you say that there was no variation. That point must be there. There was no variation. Originally, all giraffe had short necks. All snakes had no legs. You understand? There is no variation. That's the meaning. Then now they started to use the necks frequently. They use those structures, whatever question they have given frequently. And then as you use the structure, it becomes longer and bigger. You understand? And then they pass this gene to the next generation. So when you're explaining it, make sure that you use the law of use and the law of inheritance of acquired. No. And the law of inheritance of acquired characteristic. Is it clear? That sometimes it is the law of use or sometimes the law of disuse. If you want to remove that structure, then it's law of disuse. Those organisms did not use that structure frequently. If it is you want to develop the structure, then you tell us that this structure, those organisms use the structure frequently. Please, please, please know how to play. So the difference, organisms here, uh, there was no variation, there is variation at the beginning. Here we say that organisms don't change because they want to change. Organisms change because they don't want to change, because they want to change. Here in Darwinism, organisms change because of natural selection. So you have to know those things. Ne? Here, one individual in a population change. If you want to become light skinned, only you, not the whole population. But in Darwinism, if they say COVID is here, what is here, They say put on a mask, everyone must put on a mask. You want it or not, you have to put on a mask. So all the whole population will change. The whole population what? Change. But here only individuals will change. So guys, please, 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 you know have to know that. So natural selection, how it operates, please, you have to know organisms produce a large number of offsprings. There is a great deal of variation among these organisms. Some have favorable characteristics and some do not have. And then when there is a change in environment, so you have to tell us which change is it. Ne? Some have favorable character. You have to tell us which favorable characteristics is it. Do you understand? How did the tiger came up with the stripes? There is a large number of tigers. Yes, there is those which okay, we say that tigers produce a large number of offspring, isn't it? Cool. Okay, some these tigers have there is variation among these tigers, those with stripes and those without stripes. So you talked about the variation. So what are the stripes used for? Maybe to hide from the predators or to hide so that the predators don't see them, whatever, depending on the scenario they have given you. So those with the stripes, they are able to capture. They were able to hide and capture the prey, so they survived. Those without stripes, they were being recognized by the prey, and then they couldn't get what to eat, and then they died. This is called natural selection. Those which had stripes, they reproduced in large numbers and they passed their gene to the next generation. Then after that, there was a very large number of individuals with the stripes of tiger. You understand? Or after many generations, all tigers had stripes. So you explain based on the scenario. Mm. This one, punctuated equilibrium and gradualism. You need to know that in punctuated gradualism, there is check. It requires a lot of time. Ne? It requires a lot of time. Check the time it took from here up to here. While in punctuated equilibrium, check, it just changed from this. So it was here, it changed very fast. So it remained there without changing. So when you see it like this, then automatically that's punctuated equilibrium. It has periods of no change and long periods of rapid change. Periods of no change and long periods of short periods of rapid change. So you have to know that. So this punctuated equilibrium, gradualism, it requires a lot of time. It has intermediates. You see, here we now have no intermediates, which we call the punctuated equilibrium. Lastly, on this but not the least, is artificial selection. Please don't confuse artificial selection with the genetic engineering. Artificial selection, we don't manipulate genes. We only give those ones which have desirable characteristics a chance to have sex so that they're able to reproduce. Yes, I hope guys we are together, ne? And everything is good. You are hearing me properly. So we give a chance those which are able to produce. They are able to produce so that they are able to they produce. Yeah. Uh-huh. So and remember that it depends on the desire of the human. It depends on the desire. Because what this farmer might need milk, the other one might need meat. So you give a chance to those which the farmer wants to develop. So please, 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 you have to know how that operates. What are the difference between natural selection and artificial selection? Natural selection, the nature is a selecting force, while humans here is a selecting force. You understand? Yes. Selection is done to be able to adapt in the environment. That's natural selection. While here we do it so that we satisfy human needs. That's what I told you, that depends on the desire of a human. And then this one occurs within species, while this one occurs in one or more species. You go bring this and then you interbreed with this, you see? Yes. So please, 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 you have to know that. This question to write exactly the way it is here, it depends on if the question is open or the question requires you to apply. Guys, you can't go to the paper without knowing these things. Don't make a mistake. Please. Do not make a mistake. You can't go to the paper without knowing this. Hmm. There are so many people who are asking me, do I do physics? Yes, I do physics. I do physics. But I only do chemistry. Ne? I don't do physics. I don't do chemistry, physical science. Ne? Yeah. So I only do paper two. Yeah, there is another one who does paper one. Yes, in the studio. All right. So you're saying that a population of particular species, you mentioned the particular species. You understand? Ne? Becomes split into two. Yes, by a geographical. You have to mention which. Sorry, you have to mention which geographical barrier is it. Let me show you this example. I think I have an example here. Yeah. How describe speciation of potters and lemurs. Or we can even bring these bobo plants or what? Eh? Bobo plants or yes, whatever. We can bring it depending on the desire of the examiner. Describe how these they became new species. It means that there was a common ancestor somewhere, common ancestor. Hmm? So they are separated by the sea due to continental drift. Yes. And then what happened to them? These two, they are not able to interbreed, meaning that there is no gene flow. They experience different environmental conditions from here and from here. So natural selection occurred differently. If it occurs differently, what is happening here is not what's happening here, and they are not interbreeding, then automatically what's going to happen is natural selection occurs independently, and then now they're going to become genotypically and phenotypically different. And then now, when you remove this barrier and you let them mix, they cannot interbreed. Don't stop there. They cannot interbreed to produce a fertile offspring. Therefore, now you tell us, therefore, this it formed new species of potto and lemurs. So you have to mention those species they have asked you to form. You understand? Then they form a new species of pottos and lemurs. Yeah. You can't go to the paper without knowing these things. Please, 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 please. Yeah, you can't. Okay. Then we go to the next question. So this is the different methods in which species are separated. You can be separated by different breeding at a different time of the year. You can do courtship behavior. The way how you do courtship is not the same way how dogs do courtship. However, humans, you are different. You like different ways, styles, what? Hey, humans, hi. All right, whatever. This is life science. So different coach behavior. When the frog is doing, it's nice for them. It's not nice for you. So different courtship behavior. You want to go for a frog? We said humans. Right? Then number two, different adaptation of potential agents. Sometimes you can even have a baby, and you produce an individual who is infertile. So it's a different way how you can separate yourself. Lastly is the present day evolution. Present day evolution works with the natural selection. It doesn't work with speciation. You guys, you have a problem of identifying which one is speciation, which one is natural selection. It works with the natural selection, not speciation. Is it clear? Mm. So, meaning that here, before you apply, you'll find out that mosquitoes, DDT is just an insecticide. There is variation among these mosquito. So when you apply this DDT, some are resistant, some are non-resistant. So the DDT is gonna kill most of them which are non-resistant. You understand? So it's gonna remain with the resistant one. Meaning the resistant one, they're gonna survive and they pass their genes to the next generation. You understand? Then when you apply again the same dose, you'll find out that, oh, the mosquitoes are not dying. Why? Because now only resistant one are the one which survived. That's why when you go for medication, they give you different medication so that this one which is resistant to this one are killed, but the one which are resistant to this might not be resistant to another medication. And if they give you the medication, most especially antibiotics, make sure that you finish it, or otherwise you are creating monsters in your body. You're creating what? Monsters like this. If you're having HIV, make sure that you take the medication, or otherwise you are creating monsters in your body. And take it properly so that the resistance does not build. Because once you build this resistance, then you have to change the drug and you have to go for a stronger drug than before. I hope I'm clear. Hey, by the way, I'd forgotten to remind me. Don't forget to subscribe, like, and share. All right, guys, let's go to human evolution. All right. Ish. Human evolution, guys. Uh, are we there? Hey, someone was asking. I didn't see it. Yo, I feel like crying it is a lot. It's not a lot. Kumelel, it's not a lot. If you have been with together, it's like we are revising together. So now it's very easy for you to do. Just do some few questions. Don't worry. Keep on studying and practicing, sir. Make it. Yes, 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 yes. Thank you so much. Yeah, Mr. Goal. The goal, eh? Remember the goal. You have names, ne? Ah, yeah, 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 yeah. You are in Western Cape. You are listening to our twin teacher. Ah, yeah, you guys. Thank you so much, guys, for being together. Okay, classification of humans. What you need to know here is where the family to which humans belong is Hominidae. Please make sure that you know that. And then genus Homo, we're gonna see how do we use it, when and how. Know how to write a scientific name, capital letter, and then small letter. That's it. We go to the next one. This one I'm not going to delay a lot. Why? Because here they're saying upright posture. When it comes to this, don't even stress about this. Just look at yourself. I have the eyes are in front. I have my opposable thumb. I have nails instead of gloves. I have the free rotating arms. So you talk about yourself. About yourself, man, in the paper. I always tell people that it's on life science where you can copy an exam. Someone say, how? Someone is invigilating you and asking you characteristics humans share with other primates. Then you look at him that, oh, he has eyes in front, man. Yeah, you talk about things you see. And Max, on the paper, you copied from the invigilator. You guys, yeah. Life science is good. You talk about sexual dimorphism. You look at the female who is invigilating you. Yes, you can see this is the male, this is the female. You see the boobs there. Don't call them boobs. Yes. This is a female. This is a what? So you look at what you see. Yes. So don't be like raged with eh? Hey. Let me go to another section. Leave it. Then we go to this one. A difference between humans and other, in terms of cranial capacity. And they can ask you that how is cranium linked to intelligence? The higher the cranium, or the bigger the cranium is the brain box. The larger the brain cells, or the bigger the cranium, the more the brain cells, the higher the intelligence. So the cranium is big compared to the body size. That's why I say that cranium encloses. So human have a larger cranium, more than meaning that more brain cells compared to the body size. You understand? Yes, and then which suggests a higher intelligence. So guys, we have a higher intelligence because we have a high number of cells, yes, compared to the other organisms. And this is linked to the body size. I think I'm clear. Someone is saying, sir, yes, sir, you will cry. You, oh, all right, you'll try. All right. Non pomerero, ne? Mm. And then I'll look at this scalp. Eh? Guys, you got to say that if you don't know, touch yourself, feel yourself, feel yourself, feel yourself. Eh? Flat face. The other one is sloping. You just do the opposite. Flat face. Then you talk about foramen magnum in forward position. You will say forward position. We don't use any other word. Forward position to allow the spine to connect vertically. You talk about what you have. Wabona. You talk about what you have. Small canines, the other one has large canines. You understand? Large cranium, the other one has small cranium. So you look at this, you don't have broad ridges, it has broad ridges. If you are using protruding jaws, the other one is non protruding jaws. Or you can say prognanthus, non prognanthus. So it is easy. Mm. Uh-huh. Then you're saying that you're saying that so these are the differences, guys. Please make sure that you know that, né? Yes. What is bipedalism? It's what I wanted. Pedalism is walking on two hind limbs or walking on two legs. Cool. What are factors? The first one is position of foramen magnum. The second one is the pelvic girdle. The second one is the pelvic girdle. Yes, the pelvic girdle. Then the third one is the shape of the spine. Is it clear? Shape of the spine, pelvic girdle, and the position of foramen magnum. Foramen magnum is in the forward position. Allow the spine to connect vertically. Guys, it has to be that. Ne? Yes. It connects vertically. Pelvic girdle is short and wider to support the upper body weight. The spine is S shape. Allow flexibility. Look at your gogo, your mukulu. The spine is no longer a shape, it's bending like that. That's why walking is like that. When you tell him to stretch, you want to say, hey, you say that my son, man, I'm feeling pain. Why? Because the spine is not flexible, it's no longer a shape. And this is the problem tall people they find when they are growing up easily. Why? Because the spine can't support the upper weight. So what they do, it becomes a problem. Yes, meaning that they have those back pains. So this is the difference between bipedalism and quadrupedalism. Foramen magnum forward position, foramen magnum backward position. S spine shape, C shape spine shape. Short and wider, long and narrow. So you have to know that. What are advantage of bipedalism? Don't still go to the book. Check yourself, the person who is invigilating you. When he's standing, he can see at the back and in front. You can see far. That's one example. Standing upright, you helps you to see the wide field of view. Is that difficult, guys? No, man. Isn't it? It's not. When he's walking, you see even he's checking the hands. Free hands so that you can carry other things. You see? Movement is made easier and energy efficient. What if you're walking on four limbs? Is that energy efficient? No. The body is raised. So, guys, you explain based on what you see. You don't need to stress yourself with this so-called human evolution. You can't fail human evolution when you are a human. You can't. Human evolution. How? At least you fail the evolution by night selection, you flies and stuff. It's not about you. You are not a fly. Cool. But human evolution, I engage. Never. You can't. You can't. Relato, you are starting me now, ne? Relato, you are starting me. I'm not the one who said those things. Anyway, they can give you such a question. Give the vis observable what you see. You understand? Hey, what you see. What you see, what do I see here? Now I don't know the blind ones. How do they answer this question? They say observable. Then they have to say what you hear. Hey, hey, questions are questions. And you find out that the person who is blind performs better than you. How? How? Anyway, that's what God has decided. But let us make sure that, sometimes they say that we are disabled. Ne? Sometimes they say that the person who is disabled is not disabled. The person who is disabled performs better than you, then it means that when you're disabled, it's not disabled. It's you who's disabled. So please, when you read, you will, when you put something, you always get what you put in. Yes. Anyway, 4 characteristics. Now here they can specialize upper limb. Look at your arm and then talk about it. Explain how increase in the cranial volume related to intelligence. The bigger the cranium, the more brain cells, the higher the intelligence. That's it. We talked about it. What is bipedalism? We saw walking on two hind limbs. Explain each of the following structure. Okay, how contribute to bipedalism? Foramen and magnum. Forward position allow the spine to connect vertically. Cervical short and wider to support the upper weight. Spine is shaped to allow flexibility. You see, you can do this. You can do it. Evidences. We have fossil evidence, genetic evidence, and cultural evidence. Guys, when they say evidences, you say fossil evidence. Genetic evidence, not in mitochondrial DNA. Mitochondrial DNA is an example of genetic evidence. Cultural evidence, use of tools is an example of a cultural evidence. I think I'm clear there. Yeah. See. All right. Fossil evidence. What is it? You need to know what is a transitional fossil. Unfortunately, I'm not live. I would have showed you using the skulls, but I'm not live. Ne? But I did a video on this. So transitional fossil, this and this. So this becomes the transitional fossil. It has characteristics of this recent ancestor and this next what generation. So it is in transit. It is moving. It's moving from here to there. Transition fossil. It has both characteristics of the ancestor and so when you're talking about transition fossil, you have to compare. Actually, they are looking for comparison. This one, I'm not going to teach it. You have to know this table. But the only thing which I'm going to emphasize on is which one is a transitional fossil is Carabo. Which species first used the tools? It is Australopithecus. No. It's Homo habilis. Yes, is Homo habilis. First used tools. Yes, which you call the handyman. Homo habilis. And which species first walked on two limbs? It is Homo erectus, which means erect. Erect. Erect what? Standing upright. That's the meaning. Upright walking man. Erect. Homo erectus. You are walking when you have erected, meaning that you are walking upright. Think I'm clear. And don't have that in mind. Ne? I know you. You're saying that name three Sauropithecus africana species you know. Sauropithecus africana species, Taom Chad, Mrs. Place, little foot. Taom Chad, Mrs. Place, little foot. That's it. Don't go to the paper without knowing at least Sauropithecus africana species. Species which we discovered in South Africa. Sauropithecus africana species, all of them were discovered in South Africa. Wabon. Yes. Australopithecus sediba, still South Africa. So when they ask you questions, you must know these questions. Simple. Name two species which were discovered in East Africa. Then talk about Homo habilis, which was discovered in Oliver George, which is Tanzania. You understand? Yes. So, guys, please, 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 you have to know that. So when they bring about the out of Africa hypothesis, you have to say that modern humans originated from Africa and migrated to the rest of the world. This word modern, it must be there because Homo erectus, Homo neanderthals, Homo naledi, all those are humans. But Homo sapiens are the modern humans. That's why you add the word modern humans originated from Africa and migrated to the rest of the world during the Homo erectus period. Yeah, that's it. So when they ask you the evidences, you talk about fossil evidence, genetic evidence, cultural evidence. But how do you write it? This is where the problem is. Fossils of Adipithecus was only found. The word Adipithecus. Why? Because it is the oldest. Adipithecus, Australopithecus were only found in Africa. Then these ones were not only found in Africa. That's why we say that the oldest fossil of Homo erectus and Homo sapiens were found in Africa. The oldest were found in Africa. These ones, the oldest were found in Africa. But fossils of Adipithecus, Australopithecus, and Homo habilis were only, this word must be there, were only found in Africa. But when it comes to oldest, this word oldest, it must be there. When it comes to Homo erectus and Homo sapien, take a screenshot of that. Then we continue. Don't say, good day, sir. Is good night. Good day. Is it still a day? Good night. Hi. Okay, good day. How are you? Maybe you are in Dubai. All right. Anyway, right. Let's continue. Wait, good night or good evening? I don't know. Hey, English, leave me alone. English is not my mother tongue. I know that's what you usually say. All right. Mitochondrial DNA, then you have to say that inherited only from maternal line, and then analysis of these small mutations, yes, show that the oldest female, yes, ancestor were located in Africa, meaning that all these humans descended from that, which we call the mitochondrial Eve, meaning that Eve was from Africa. In other words, they are saying these people that Eve was a black person. Why? Because in Africa we find black people. But are they black? They're not black, man. They are dark-skinned people, not black. Anyway, you can't call them the way you want. Yeah. So you're saying that they're like a pot. They are black. Anyway, so when they ask you colors, you say that's black. Ne? You know this Moses. This one. Others they call them colorless. So when you're colorless, they're skin light skin. Me call them that. But anyway, if I'm black, I'm colored, guys. If you are black, you are colored. If you are white, you are colored. So everyone is colored. Yeah, they just want to make us divided. Aye, man, we are all of the same because we have the same type of blood, red. Anyway, I'm not talking about that. Let me just keep it quiet. Yeah. The oldest tools, you know them, they are called Order One tools. Ish. Guys, I think I'm supposed to be closing now. But anyway, let me just do this highlight very fast, so that you don't fail that phylogenetic tree. All right. We have different perceptions, Don't take me wrong, I was just talking. And I'm not that kind of guy who appreciate everyone. Anyway, let's start. And then we continue. We get our distinction. We smash the paper on Monday. What is the name given to this diagram? This diagram is called a phylogenetic tree. How many genera are shown in this diagram? When they say genera, guys, look critically and look here. Uh-uh. What's happening with my screen? Don't tell me that. Uh-uh. So it means it happened there. All right. How many genera? So you look at the first one is Adipithecus. Remember, genus is the first name. The second one is Australopithecus. This one is Australopithecus, so I don't repeat. So you have two Paranthropus. Those are three. You see, Paranthropus, Australopithecus. I don't repeat. The fourth one is Homo. One, two, three, four. Do I have another one? No. I think there are four. So there are four genus. That's how many genera. Is it clear? Yes. Yes. So how many genera? I think I've answered that. What is the common ancestor of Homo and the Homo sapien and the Homo erectus? You come here, Homo erectus is here, Homo sapien is here. When you join them, they join here, meaning that the common ancestor is Homo ergaster. Based on this diagram, is Homo ergaster. What is the common ancestor of all species? Automatically is Adipithecus remidus. Give two pairs of species which competed for resources, meaning that give two pairs of species which existed at the same time. So it means that you just have to draw, just choose. Maybe I'm gonna choose this. Draw until it touches. So you see this, it's in the same line, even this. Yeah, they exit at the same time, but it could be half of the time of all the time. So, but they exist at the same time. So if they exist at the same time, it means that they competed for resources. Hmm. Then they're saying that which species first use tools. In other words, it is Homo habilis. But based on this, you have to look at whether they gave you what to use, the scenario or not. Based on this scenario, the tools are here. They're saying that, yeah, the tools started here. So maybe it is the Paranthropus Ethiopicus, the one which first used tools. Then they're saying that which species first walked on two legs. Here we talk about two legs, two legs, two legs. Two legs is bipedalism. So you look for bipedalism. Bipedalism is this one. So it's around there. Which one is it? Yeah, Australopithecus and Amensis. You see? So that's how you identify. Then they are saying that mhm. And they're saying that which species first walked on, okay. How many million years ago did Australopithecus africanus appeared? Australopithecus africanus, I look for Australopithecus africanus, I think it is here, then I calculate. It appeared around 2.9 million years ago. And then when did the Homo habilis became extinct? When did the Homo habilis became extinct? Homo habilis, Homo habilis, Homo ergaster, Homo neanderthal, Homo habilis is here. It got extinct around 1.6 million years ago. Then they're saying that how many million years did Australopithecus africanus existed? So it means that you have Australopithecus africanus where it started and where it ended, then you subtract. Yes, then you say million years. Here you don't say million years ago because they're saying yes. They're saying how does brain size show evolutionary relationship or trend? We say that the bigger the cranium, the larger the brain cells, or more brain cells, the higher the intelligence. Meaning that these organisms will use advanced tools. You understand? Yes. Remember, we said that these tools, the more you modify them, the more they become more. The more you advance the brain, becoming bigger, it becomes the tools become more sophisticated. Meaning that these tools they require thought. You require to think to make them. Give an example of a transitional fossil. We said it's carabo. How does dentition show the diet of the species? Large canines meaning that they are feeding on rough food, and then the smaller piece they show that they are feeding on forked food. Guys, I think that is it. We'll stop here. Thank you so much. Tomorrow, I think I'll make a cross night. It will be a cross night. Please, please, please make sure that you go through this video 3 times before you go for the paper. I repeat, make sure that you go through this video 3 times before you go to the paper. When you come out of the paper, call me or tell me here, what does it mean? with me saying go through it 3 times. You'll understand this when the paper has come. Is it clear? I wish you the best, and I know that you guys, you gonna make it. I want you to be responsible citizens. Ne? Please, please, please be responsible citizens. Now you can have a relax, you watch small TV, then after that make your brain relax, and then you come back and redo, or you watch the video, or you revise. Ne? Yes. See you again when we come back for the last time. Relato, you are welcome. Hey guys, sleep well. I see you, Sandy Le. I see General. You have DJ AJ Junior. All right, Rivonia. I see you guys. It has been a nice lesson. I'm seeing Doomsay. I see you, big boy. Big boy, thank you for being with us from the beginning until the end. Rivonia, you also did that the same. Thank you so much. I see you and I salute you. I wish you the best, guys. Doomsay, please go back and then replay the video. I explained DNA. Thank you so much. See you again. It has been M. Saidi. As usual, don't forget to subscribe, like, and share.